Mitosis and Meiosis Project by Mike Cutler and Anna Peters.
Mitosis and meiosis are two possible ways that a cell can divide during the cell cycle. The cell cycle can be broken down into three main events: interphase, cell division, and cytokinesis. Interphase, the longest of the three events, has three sections: 1, the first gap; 2, the synthesis of DNA; and 3, the second gap. The stage next in the cell cycle is cell division. In this stage, genetic material is divided. The stage following cell division is cytokinesis, which is the division of a cell's cytoplasm. Together, cell division and cytoplasmic division make up the mitotic M phase.
The importance of mitosis is to create two cells from one genetically equivalent parent cell.
Mitosis occurs in a series of phases. Before mitosis occurs, the cell is found in the G2 stage of interphase. In this phase, the nucleus is enclosed by the nuclear envelope. One centrosome duplicates to form two centrosomes. Chromosomes are not condensed, and a nucleoli is present. During the next stage, prophase, mitosis begins. Chromatin fibers begin to condense through cohesions. The nucleoli disappears. Duplicated chromosomes become sister chromatids. A mitotic spindle is formed that begins to move away towards the poles of the cell. Prometaphase occurs when the nuclear envelope dissipates and therefore allows microtubules to move into the nuclear space and connect with the kinetochores of the sister chromatids, causing some to move back and forth between the two poles. Metaphase occurs after prometaphase is over. In this phase, the duplicated centrosomes are completely opposite of each other. The chromosomes are aligning on the metaphase plate, and the kinetochores are all attached to a microtubule from the opposite side. Anaphase occurs when the tight cohesions between sister chromatids are broken and the cell elongates, causing the number of daughter chromosomes on each side of the cell to be equal. The final phase of mitosis, telophase, occurs when two nuclei form from the daughter cells on each side. Nucleoli reappear, chromosomes become less condensed, and a nuclear envelope reforms from the remains of the parent cell's nuclear envelope. This is the last stage of mitosis. Cytokinesis occurs after this when the cytoplasm is divided between two cells, either by a cleavage furrow found in animal cells or a cell plate found in plant cells. The final result of mitosis is two diploid cells that are genetically identical to the parent cell.
Mitosis ends with two daughter cells that have the same number of chromosomes as the parent cell. These are diploid cells, cells with two copies of every chromosome, or 2N. The cell that starts mitosis as a diploid cell ends mitosis with daughter cells that are diploid and are genetically identical to the parent cell.
The importance of meiosis is to create 4 haploid cells for every one diploid cell. These cells are used for reproduction.
Meiosis begins with prophase 1. In this stage, chromatin condenses into chromosomes, and paired homologs become connected through synapsis. Crossing over may occur, resulting in chiasmata. The spindle apparatus forms and the nuclear envelope breaks down. Next is metaphase 1. This is when the homologous pairs of chromosomes align at the metaphase plate. Microtubules from each pole connect to the kinetochore of each chromosome. Anaphase 1 occurs when homologs separate and they move towards opposite poles, being moved by the spindle apparatus. Finally, telophase 1 happens as the chromosomes have now moved to opposite poles and a nuclear envelope forms around them. Cytokinesis typically occurs within telophase as two haploid cells are formed.
The main goal of meiosis II is to separate the newly created sister chromosomes. In prophase 2, a spindle appears in each of the two haploid daughter cells, and chromosomes start to move towards the metaphase plate. Once on the metaphase plate, metaphase 2 begins. Due to crossing over in meiosis I, sister chromatids line up with their non-identical homologous sister chromatid. Anaphase 2 occurs when the sister chromatids separate and move towards opposite sides of the cell. Telophase 2 then occurs when a new nuclei is formed and chromosomes decondense. This is then followed by cytokinesis, resulting in an end result of 4 haploid non-identical cells, which originate from one parent cell.
4 haploid cells are formed containing half as many chromosomes as the parent cell. Each cell is genetically independent from other cells.
Mitosis and meiosis have both similarities and differences. Mitosis is a somatic cell, while meiosis is the gamete. The result of mitosis is 2 identical diploid daughter cells, while meiosis results in 4 non-identical haploid daughter cells. Mitosis also has 1 division, while meiosis consists of 2. The similarities between these two types of divisions are that they are both ways that cells divide, and DNA replication occurs during interphase in the S stage.